Hello everybody, great to have you here. Thanks for joining. Welcome, please say hi in the chat if you're here, especially if you're new. As always, let us know where you're coming from. Joel, good morning. Thanks for being here. Today we're going to talk about genome assembly and evolutionary trees. This will kick us off with probably quite a few problems, especially in the evolutionary tree side of things. And we're going to start with some introductory problems today. Well, we're also going to see some questions that at first glance are going to seem really challenging, and we're going to see an example of something where sometimes we can answer a question much faster than we might imagine if we just use a little bit of math. So you might be scared of math, but I love math, and hopefully I'm going to be able to convey that sometimes it's actually really useful for helping us to understand things and to answer questions very, very quickly. If you're following along, we've got starter code and the completed code for this video in the description, so check that out there. If you have any questions, as always, let me know. So just chat any questions that you have. I'll be tracking those. I usually answer any questions on this stream in between problems that we solve. So let us know if you have any questions as we go. Henry, welcome. Anna, hello. Great to have you here. Leonardo, welcome. Our announcement, or a little bit of news coming this week, in our partner course, Programming for Lovers, we're finishing up. We've got a few more weeks of streams, but we're getting to the end of our long journey. But it's not really the end, so we're going to have opportunities to complete projects and check out our partner course for that. You can do a project on sort of introductory programming. You can do a project on more advanced programming. And one of the things that we're looking at right now is the opportunity to, once you have the foundations of programming, which a lot of people have, whatever that means, you have some exposure to programming, whether it's my course or something else. It seems like a lot of folks are interested in, how do I effectively build something faster and bigger using AI as a guide? So we have let's call it like a soft interest Google form that you can fill out. If you're interested in that, if that sounds like something that you would be excited about doing in the coming weeks or months, fill out our form. I just dropped a link to it in the chat, and you can check it out and let us know. So you can give us your email or just let us know sort of like what your general interest would be. If you're not on our mailing list, then make sure that we have your email if you're interested in that. And I think that's a great reason why you should be on our mailing list. So please make sure you like and subscribe to keep us visible here. But if you're interested in hearing about a lot of the other stuff that we're building out, including like our AI project, make sure you're on our Discord, make sure you're on our mailing list. It's the best way to stay in touch. And that's all I have for introductions to the stream today. We're going to solve 5 problems. The problems we're going to solve are, as always, we put those in the description so you can check those out. The first couple of questions are going to be around the start of genome assembly, and then we're going to look at a couple of questions, or 3 questions, around evolutionary trees. So we're kind of starting both of these ideas. We've worked around a lot of this stuff before. We've started talking about how do we compare strings. That's going to be really helpful in both of these contexts. And we've seen some advanced dynamic programming algorithms for that. Today we're going to start off, what does it mean to put a genome back together? Look at the very basics there. And then with evolutionary trees, what do we mean by a tree? And what is it that we're ultimately trying to do when we build an evolutionary tree? So we're going to see some kind of starter problems in that area too. So again, if you have any questions, let us know. We're happy to take a look at those as we go. Otherwise, I'm excited to get started. And we're going to start with the question or the problem with ID long. And, I'm not actually sure why it's called long, but it is, and that's where we're going to start. So let's code. All right. And I should say we're going to do long, CORR, TREE. That one makes sense. It's going to be a tree. This one is called INOD. And then the last one we're going to do is PDST. But we're going to start with long. All right. LONG is genome assembly as shortest substring. And this problem is built on a figure that I think is pretty much somewhat of it's been the same picture since the 1970s, which is that at least when we talk about what's called de novo genome sequencing or genome assembly, it relies on this paradigm of fragment and assemble. So you can easily capture a lot of DNA. In your case, for example, we can sequence your genome by getting a blood sample and then isolating white blood cells and isolating the DNA from the nuclei in those white blood cells. And you got a lot of sample. No one has ever been able to come up with a technology that will read out an entire chromosome of DNA, though, tens, hundreds of millions of nucleotides at a time. The best we can do is to fragment our DNA and then feed it into a sequencer that reads out short fragments. Then the idea is, well, because we started with multiple copies of our original genome, you can then start overlapping the fragments in order to infer what the genome is at the end. And we want to assemble those fragments in what we might call the most parsimonious way, meaning the simplest imaginable explanation. In this particular case, we can frame a very introductory version of genome assembly by saying, well, say as our input, we get a collection of strings, and this collection of strings is going to have a very precise set of guarantees that aren't going to be tremendously practical, and that's okay. We can come back to practical directions in this field later. But the idea is we're going to assume that we get a lot of nice overlaps. So we're going to give at most 50 DNA strings, so it's not going to be a huge data set in this problem. But there's a condition that we know is always going to be followed, which is that there is a unique way to reconstruct the chromosomes, or the sequence that they're coming from, by gluing together pairs of reads that overlap by more than half of their length. So that means if I have a DNA string, and I ever find another DNA string that overlaps it in half of its length, I know I can join those two together and start to join them into a longer genome. That's a strong assumption, but that means that what I can do is follow a greedy algorithm that will reconstruct this, because what we ultimately want is a superstring, meaning a string that contains all of our input substrings, or strings as substrings, that is as short as possible. And because of this condition that, there's always only one way of gluing them together, meaning we're guaranteed to have, for any one of our fragments, or what are called reads, for every one of our sequencing reads, we know that there's always some other read. If we're not at the end of the genome, we know that there's some read to its right or to its left that overlaps in half of its length. That's good. That means if we follow this greedy algorithm, we're going to get a superstring of our input collection of DNA strings that's as short as possible. And we want it to be as short as possible because of this notion of parsimony, which appears throughout biology and throughout science, and is also often called Occam's razor. But the idea of, if we have a lot of explanations for something, we want to take the simplest one. So in the case of genome assembly, if you have a collection of data, you want the smallest genome that explains all of those sequencing reads. So a greedy algorithm here is what we're going to apply. We're going to look at every single string, and then we're going to try and say, can I find another string that overlaps it to the left? Can I find a string that overlaps it to the right? And if I can, and it's half of its length or more, then I'm going to start continuing and iterating on building this string as we go. Now, the hint here is that this algorithm is very bad and scales very poorly. We're not going to have 50 DNA strings in practice. We're going to have hundreds of millions of sequencing reads for a human or eukaryote like us. But all of this is a story for another day. So we're just starting with getting comfortable with the paradigm here and understanding that what we care about is overlapping fragments. That when at the end of the day, the only way we can reconstruct a genome is by identifying that some of these fragments overlap with each other. And the purpose of this problem is just to introduce us to that paradigm and get us comfortable with it. And to say, let's see what happens when we try to build a greedy genome assembler. We'll get more into better algorithms here, and we go very deep into this in our bioinformatics algorithms project. So if you're looking for, a deeper treatment that sort of brings you up to speed in terms of the state of the art and graph structures for doing this, that's a great resource to consult. This is just our warm-up question on genome assembly for Rosalind. So that's what we're going to do. We need a function called shortest superstring. So if you look at our input and output, we're going to get a bunch of FASTA strings, and we're going to want to assemble them in this shortest possible fashion. So you see this string, for example, appears to be at the start. And we know that it's going to overlap by at least half on the right side. And so we can actually see that here. So we see that string starts to appear here. And then the next string we're going to get is this string that starts with CECT because it overlaps with at least half here. And then this GCCG overlaps with at least half of that one that we had. So this is going to be our first one, second one, third, and fourth. And at the end of the day, when we overlap them all, we're going to obtain this final output, which is our superstring. So let's write this greedy algorithm just to kind of explore the problem. And we have a function called shortest superstring that takes our collections of reads as a list. So this is just a list of strings. And then it produces the shortest string that contains every read as a substring with this very strong assumption that anytime I find a partner that overlaps me by half of my length, I'm just going to append it to the growing genome. So when I solve this problem, the first thing we're going to do is let's create a list to store remaining reads that we haven't used. And so I could use my read set directly, but I don't want to necessarily start deleting elements from it. So I'm going to have basically a copy of the input set that we have. So I'm going to call this remaining or remaining reads. It's going to be a list of strings. It's going to start off as empty, and we can go through and look at all of our reads, and then I'm just going to append each current read to this collection of remaining reads. Then what we really want to build is our string. So at the end of the day, we want the shortest string. So our idea is let's declare a genome, which we will add to as we go. So I know I'm going to have genome equals some empty string. In fact, we can go through and say, let's just set it equal to reads of 0. And then at the end, we're eventually going to return it. So there's nothing special about reads of 0 here. It might be in the middle of the string. It might be at the end of the genome on the left or the right. We're not really sure, but we do know it has to be used. And so we're going to initialize our genome by saying, well, let's look at our first read. And then that's going to have where we start our genome. So the idea here is, again, if we're thinking about this is an introductory problem, we're using a greedy approach. We're really just kind of trying to match human intuition on how you would do this. If I gave you these 50 fragments, you might say, well, look at my current read, and I'm going to keep trying to grow my genome by saying, for the current genome, is there one that I can find that overlaps the left side? And I'm going to try to extend the genome to the left and grow it that direction until I can't anymore. And I'm going to do the same thing in the opposite direction. So I'm going to go right as far as I can. And so we're really just doing a looping process where I'm going to use a flag. So this flag is going to indicate that we keep looping, or keep looking to the right or the left. And I'm going to use it in both contexts. So I'm going to have extending is equal to true. And while it's the case that it's still true, and there's still something left in our reads. So every time I use a read, I'm going to get rid of the collection, the current read that we have. What I want to do is look to the right. So I'm going to call a function called find next read right, and I've written that here as a subroutine. It's going to take the genome and it's going to take the remaining collection that we have. And what that is going to return, and we can look at what we have here, is it's going to return three things. It's going to return the read itself. So if we find something that can extend us to the right, that overlaps with the genome to the right, it's going to return the index of that read in the collection that we're working with so that we know where to access it. And then it's also going to give us the overlap here, which is, and you're going to say it's a string, it's an integer. What is the overlap? It's the, sorry, I'm going to even expand this out, where read is the read, index is its index in the read set, and overlap is the number of symbols that overlap between this read and the input genome. Okay, so I'm just specifying that a little bit more clearly. So this is, in other words, some number k. That will help us because when we find the overlapping read, we need to say how many symbols does it overlap in, and that's going to allow us to know when we're sliding it where it is I place it in the growing genome. So we've got our find next read right. We know it's going to return these three things. It's going to return the next read that I care about. It's going to return the index, and it's going to return the k, which is the number of nucleotides that they overlap in. Then we're going to say if that index is equal to -1, that's our default case, and this index equal to -1 is going to be returned when there is no read that we find. So that's telling us that we didn't find anything to extend it. And that's when I'm going to stop this process. So I'll stop the process by just making sure that I set extending equal to false. And then otherwise, in this case, we found an overlapping read, and that's good. So in that particular case, I'm going to say, well, I've got the next read, and I've got the current genome that I'm forming. And I know that it overlaps with the current genome in 3 positions. Or 3 positions, in k positions. You might wonder, why did I say 3? I don't know. I read k here, and that looked like a 3 to me for some reason. So if we find that, it would be as if we had A, B, C, D, the genome is equal to A, B, C, D, E, F, G, H, I, J, K. And the read is equal to H, I, J, K, L, M, N, O. Okay, so if that's the current case, then we would say, well, in this case, K then is equal to 4. And the new genome should be what happens when we take the existing genome and then we append to it everything except for the first 4 symbols. So the new genome should be genome plus, and in this particular case, it should be that next read. I think I called it next read. In this case, it should be that next read. And it should be everything starting at position k to the end of the string. So we're looking for a suffix of next read starting at position k. And look, that makes sense here because we have H is position 0, position 1, 2, 3. So position 4 starts at LMNO, and that's what we want. Okay, so there's nothing wrong with writing down just a simple example to keep us on the right track here. And in this case, when we find this overlapping read, all we're going to do is say genome is equal to the current genome plus the next read, starting at position K, going to the end of the string. That's really good because it gives us what we want. There's going to be repeated string concatenations, which is something that we've seen is not always something that we want to do, but we know that there's at most 50 reads in our data set, so we're not going to be doing a lot of string concatenations for this particular problem. So we don't have to worry about it because of the scale of what we're doing here. The other thing that we want to be careful about, though, is let's remove the read from the data set. So we also need to say the collection of remaining reads is equal to what happens when I hack it out of the collection of remaining reads. So this is where having the index of the read that we just found is going to be so useful, because we found a read, we know its index in this data set, and if I know its index, well, I can get rid of it with a kind of a classic approach to remove an element from a list, which is a cross-language idea, which is to take the sublist or the slice of this data going up to but not including this read that I found, and then adding to it everything past the read that I found. So looking at index plus 1 and going to the end. Once I've done that, then it's just a matter of saying, okay, I have extended this genome as far to the right as I can. And now we need to look left and repeat the process. So what am I going to do? I'm going to use a flag. Extending is equal to true. While that remains true, and there's still anything in the reads, I'm going to do the exact same setup. So everything here is the same, except if that's the case, okay, I think we had a little bit of connectivity there. I think I'm back. Oh, good. Okay, thanks, Jill. All right, I didn't do anything while you were away. So we're looking to the left, in other words, and our code is generally the same as what it was previously. So we have extending equal to true. While we're still looking to the left and there's anything left, we're going to look to the left. We're going to get three things: what the overlap is, how many symbols there are, what the index is, and what the next read itself is going to be. So if we didn't find anything, we're going to stop extending. If we do find something, here is where we need to say which symbols overlap. And we could look at an example, just like what we had before. So let's look at something like this. Okay, and for this, I'm going to say instead, let's start at it having a genome like that. And our next read is going to overlap here. So let's say C, D, E, F, G, H, I. And in that particular case, we're going to get an overlap of 3. Actually, let's go J, K. So I have the condition that it's at least half of its length. So if K is 5 here, well, it's the same idea, except that the new genome should be C, D, E, F, G, H, I, J, K, L, M, N, O, P, Q, R, S, T. And we get that by taking the next read first and then appending genome to it, except we're going to have some overlap here. And so we don't want the final 5 symbols of next read when we perform that overlap. So to do that, what we'll say is, okay, the genome is equal to the next read plus the genome, but I want to make sure I could do this in one of two ways. So one of the things I could do would be to say, you know, I don't want the first K symbols of genome because the next read's already going to have them. It's weird to, you know, so I could do everything starting at position K up to the end of the genome. That would be a little odd. And the reason why that would be a little odd, I think, is because, well, it feels like I'm getting rid of the genome. So instead, what I could do is to say, well, let's not get rid of the genome. Let's use the whole genome. But let's not use certain symbols of the next read. In other words, I want the prefix of the next read up to the position where I stop having the K overlapping symbols. And so I want the prefix of length, whatever the length of the next read is, minus K. Okay. So we know that this needs to be the length of the collection of symbols that I want. So in this particular case, we have next read having length 9. The overlap is 5. And so we want the first 4 symbols. So we want a prefix here of 4 symbols. So length was 9, K was 5, and the length of this substring is just this index minus the hidden 0 that's there. So that's a fast way to make sure that we have our substring indexing correct. I will, of course, not have the 0 there because it's implied. And then we need one more thing to parallel what we did before, which is we also want to remove the read from the data set. So we want to make sure we do that too. But that's the same line of code that we had previously. So we've got 2 functions to write here. There's 2 little subroutines. We need a function that's going to find our next read to the left and find our next read to the right. And otherwise, short of superstring is complete, because after we've done this, we just return the genome, which we know we've already written. Let's go to the right first. They're going to be similar subroutines, but we're going to find the next read to the right. And again, remember, we're given a genome and a collection of reads. What we're looking for is the read that overlaps the genome by at most half its length, what its index is in the read set, and how many symbols it overlaps by. So that was what we said was K previously. And this should be a pretty straightforward function. Let's range over our reads and look for anything that overlaps by the requisite length. So I'll say for I in range over, let's say num reads, I like that, and then I'll say num reads is equal to the length of our read set. And as we do this, well, the current read, I'm going to call it R, is equal to the ith read. And I will ask, you know, how much does R overlap the genome by on the right? So I'm going to use a subroutine to answer that question, and it's going to give me an integer value equal to K. So I'm going to call overlap length of genome and R. So here's our subroutine. And there is a threshold, which is, we need to say it's length of R divided by 2. And, you know, this is where there's a little bit of vomit that I have, because this is where the problem's just, you know, it's an introductory problem, but we're using a very, very strict assumption here that's not pragmatic, which is sometimes, of course, sometimes you're going to have reads that don't overlap in at least half of their length. And this is like an arbitrary hard-coded parameter in the problem. So, but it's okay, you know, that's all right. And technically, we should be a little careful here, because it might be the case that our read is longer than the genome. So there is a minor technicality, which is that I should say this is the minimum of the length of the genome and the length of the read, and then divide by 2 integer division. But, you know, if we found an overlap of a substantial or sufficient length, then we're done. Because the data set is guaranteeing us that if we find something where the overlapping length between the genome and R is at least equal to the threshold, then I'm just going to return, because it's going to be unique. So if the overlap length that I found is greater than the threshold, then I'm going to return R, I, and K. And then there's the point, which is if we make it here, we didn't find anything. And if that's the case, then I want to return something default. So what I will return is an empty string, because I was unable to find a read. The critical value that I care about is -1. That's how I will test that I didn't find anything. And we've already assessed that when we wrote shortest superstring. So when we wrote shortest superstring, I checked that I didn't find anything by having this index in the reads be equal to -1. And so -1 is going to be the index value. What should K be? What should the overlap be? Well, I didn't find anything overlapping, and so it should just be equal to 0. And then we're going to take the same idea, and we'll apply the same idea to find next read to the left. So everything here is going to be immensely similar. You might even wonder, how does this even change at all? And that means that there might be a subroutine that combines these two ideas and just has a left or right flag into a single function. For now, I'm going to keep it as 2 functions, but you might play around with that to see how you can combine these 2 functions into one. Because I'm still going to have the number of reads. I'm still going to range over that number of reads. My current read is reads of I. And I'm going to change this to left. And that's where things are going to vary. So the only difference here is that, well, I'm overlapping the read with the genome. So the order there is really important, because how this function overlap length is going to work is this is the first string and this is the second string. So I'm asking, does the first string overlap with the second string in any symbols? And that's a different question from trying to do them in the opposite way. So that's where the order given to overlap length is important. But at the end of the day, this line is going to be the same if I'm still going to be checking if K is bigger than the threshold, and then I'm going to return this and I have the same default. Now it's just a matter of writing my overlap length function. So I've got one more function called overlap length that takes 2 strings as inputs, and it's trying to return the number of symbols where they overlap. The point is, it's the largest possible value of K. So, just because you find an overlap doesn't mean it's the best one. I guess there's a simple example here of something like this, right? Where would you say that these 2 strings overlap? Well, these 2 symbols overlap the end here, right? And so do these 6 overlap the end here. But what you're looking for is the overlap that is maximal. So you're looking for, in this case, it looks like 10 symbols that overlap with 10 symbols at the end of S. So because we want the largest one, what this means is we need to check all the different ways. So we say, does the first symbol overlap here, or do the first 2 overlap with the last 2 here? So we're going to need a loop that checks all of those possibilities. And I'm going to say, we're going to declare a max check variable to be equal to the minimum of the 2 lengths of the input strings. And so what's nice about this is we can't have an overlap bigger than this, because it's not possible for 2 strings to overlap in more symbols than the length of one of them. So if one of these has length 100 and one of them has length 10, there's no way you're going to get an overlap of length 50. So max check is just going to store that minimum, and then I'm going to range over all of the possible overlap lengths. Okay, and we're going to do that by saying we're going to use a value of k to indicate that. And we're going to start at 0 and range over up to, but not including max check. And then we'll say, if it's the case that we find, does the suffix of s of length k equal the prefix of t of length k? And then if so, we found, that they overlap in these positions. So we say if s of length of s minus k up to its end, so this is the suffix of s of length k, if that's equal to the prefix of length k of t, so do this, suffix of s of length k equal prefix of t. I feel like I've got something wrong here. Yeah, okay, good. So yeah, this is the final s, length of s, this is the final k symbols of s. So we have length of s minus k is the starting index, and then we have the upper index here because this is the prefix of t. All right. But then the question is what to do. Well, we can't really take any action. And the reason why is, well, we could update the best value found so far. So what we would have to do is we would say, well, best k is equal to 0, and then best k is equal to k. And then at the end of this, we would say, what do we have? Did we ever find anything? The last thing that we found would be our best k, and I could return best k. And that's nice. That will work. But in any language like Python that allows us to very natively do the ranging in the opposite direction, what we're going to do instead is we're going to start with the biggest possible one. What's nice about that is what I'm going to say is I'm going to start at max check, and I'm going to go to 0, and then I'm going to go down by 1 each time. So this is the step length. So I'll start at the big value, and I'll go towards 0. And I want to return 0 as my default, but I'm not going to need best k. And the reason why is, as soon as I find a match, I know this is the biggest one. Why? Because I'm starting at big values and I'm going down. So as soon as I know that's the biggest one, I can simply return k. And that's our function. And we can try it out. So I can run python 3 long.py, and we've got a test data set. So we've got a collection of reads here. That's our sample from Rosalind. It gives us something that appears to match here, so that's good. We can comment out the parsing part of this program, download a data set, and I'm going to move it into our file explorer and call it dataset.txt. And now when I run it again, I'm going to get an output, and I hope it's right. And we get success. Okay. So even though this was a make-believe version of genome assembly, one where we don't have to worry about things that are very practical, like, well, what if they don't overlap in, these perfect, at least half of the reads circumstances. So what if you have fewer reads, or there's positions where you're just not getting that much overlap? What do you do then? And Henry has a question about, what about lots of repeats? So that's a great question, because a greedy approach is going to fail spectacularly on such a data set. If I have a read and I know it overlaps with another read, well, there's a good chance it's going to overlap with a lot of reads. And the reason why is this read could have come from a lot of different positions in the genome. Our genomes are 3 billion base pairs long, and about half of them are made up of these repeated regions. So that means if I get a fragment of DNA, the challenge is I don't know exactly where it came from if it's belongs to one of those repeated regions. This is what makes it such a challenging problem, because of the scale of the data, that you're going to have hundreds of millions of sequencing reads. as well as the fact that you're working with a genome that is very repetitive. All that's the type of thing we cover in bioinformatics algorithms, and it's the subject of another day. The purpose of this problem was to show, even in the case of making a lot of assumptions, we still had, a pretty reasonably long solution to this, and it at least warms us up to these sorts of considerations. All right. So we're going to look at our next question. I think, had a prerequisite of that genome assembly. We're going to be looking at error correction. So what happens when we start to peel the layers of the onion away on genome assembly? And the first thing that we're going to look at is the fact that our reads might have errors in them as well. So this one is going to be CORR. And again, if you've got questions, let us know. We'll cover them in between. All right. CORR is error correction in reads. And we've already taken this look at genome assembly with a simple but effective introduction to how it works. One of the assumptions that we've made thus far is that, when we have a collection of sequencing reads that we want to overlap and form the resulting genome from, the assumption is that the data are correct. Real sequencing machines, unfortunately, are going to make errors, and it depends on the technology that we're using. The oversimplification of the process is, you usually get 2 of 3 things that you're looking for at this point in time, and it's been the case for a long period. If you're looking for inexpensive reads, you're looking for accurate reads, and you're looking for reads that are long because you want to read as much DNA as you can with each fragment, you get to pick 2 of those things. The most popular sequencing technology chooses accuracy and inexpensiveness, but different technologies will get you 2 of those 3 things. Even in a technology that gives you accurate reads, though, accuracy usually means, maybe you make an error on the order of 1 out of every 1,000 nucleotides. Other technologies sometimes might give you very long reads, tens or hundreds of thousands of nucleotides long, and if they're inexpensive, they might actually only be correct in about 85% of the nucleotide positions. So when you get your data, you can only trust about 5 and 6 nucleotides that you see. The assumption of this problem is going to be that we're working with much more accurate data, where you're usually only going to be seeing a single symbol error relatively infrequently. And what the problem is getting at is, how is it that we might go about the idea of processing the data before we start to work with it and decipher where there might be some errors hidden in it? Because this is biology, if it can be hard, it will be hard. So there are other reasons why it might be the case that when I sequence my reads, I see one read and I see another read that's similar to it that maybe differs from it in a single substitution. One example would be, this could come from a repeated region of the genome. It could come from a repeated region where maybe there's a single nucleotide change in this repeat. That's a commonly occurring phenomenon. And the other possibility is, if you're sequencing, say, a eukaryote like us, where you have pairs of chromosomes, homologous pairs, and you have one chromosome in each pair inherited from your biological mother and the other coming from your biological father, most of the DNA is going to be similar, but every so often you're going to have a position where one parent has like an A and the other parent has a C in this position, or whatever the 2 nucleotides are. But because of these single nucleotide changes at like these individual positions, you're going to see that there are going to be reads that often have this phenomenon because of that as well. So this is a much harder problem than it might initially seem. That, well, we look at our data and then we say, can we find within our data pairs of reads that are kind of close to each other? That doesn't necessarily only imply that there is an error there. That also is a subject of another day and the subject of a much longer discussion in our bioinformatics algorithms project. But much like in our introduction to genome sequencing problem, the idea of this problem is just to get you comfortable with the idea that, well, we could, in theory, start to error correct our reads before we feed them into an algorithm to try and assemble them, to try and identify places where there might be errors, or there might be repeats with small changes, or there might be this phenomenon that I mentioned before of having the same position in two parents differ, or heterozygosity. So this particular question is just looking at, let's take a collection of strings that are all going to be in this FASTA format. And for every one of the reads in our data set, we're going to make an assumption about the data set so that we can have, it's going to be a strict, unrealistic assumption, but it's one that allows us to get a flavor for what we're doing here. And one of the possibilities is that the string was correctly sequenced. And if it is, we're going to make this hard assumption that the data set contains this string at least twice. There's a note here that it's possibly as a reverse complement. And the idea there is that, well, DNA is double-stranded, and we need to take reverse complements into account. So when you sequence a position of the genome, it may be the case that we sequenced it from one strand of DNA, or it could have come from the opposing strand of DNA. And so the problem is attempting to model that. And then the second assumption of what could have happened is that the read is incorrect, appears in the data set exactly once, but there is a partner for this read that is almost identical to it, keeping in mind this idea that these errors are something that is always going to happen, but are relatively rare, maybe 1 out of every 1,000 nucleotides. So we're going to say, well, let's make the assumption that if it is incorrect, it has Hamming distance 1, meaning it only mismatches in one position with respect to some partner in the data set. So the idea here is we're pre-processing the reads to find, are there collections of reads that are similar to each other? And that's a compelling, important thing to do. The problem is just making some hard assumptions, or some strict assumptions, I should say, about how it is that we are assuming the data are given to us. So the point of the question is to say, can we perform this error correction? Meaning, can I say, we've got an old read, and we're going to correct it into a new read. So each correction has to be a single symbol substitution, and it may be that when we make that substitution, it matches its partner, or it may be that it matches the reverse complement of its partner. So there's a fair amount to do, even though we're making this simplifying assumption. And I will say, let's look at our example. We've got a collection of fragments here. And the collection of fragments, we have 9 total DNA strings. If we look at, say, the second read, it's one away, so TTCAT, it's one away from TTGAT, which is our 8th read. Another example would be read 9, TTTCC, is one away from TTTCA, which is our 6th read. And we could just as easily have said TTTCA is one away from TTTCC. So in our corrections, the autograder is going to allow us to do either one of those directions. We could correct this one, or we could correct this one. We could correct this one, or we could correct this one. We don't know which one is incorrect and which one is not, but the idea of the problem is just to pair them off. And then finally, we have GAGGA, which is our 6th read in the data set, or our 5th read, I should say. And GAGGA doesn't have a partner associated with it, but if we change it to GATGA, the reverse complement, so if we look at the reverse complement of this thing, the reverse complement of this is TCC TC. And if we look, we can see that's actually one away from TCATC, which is here. So the reverse complement of TCATC is GATGA, which is close to this. So it's just missing that middle symbol. Okay, so our idea then is let's range through the data set and let's find everything that occurs twice, possibly with reverse complements. And anything that doesn't must be an error. And so we need to find the partner associated with it, also with reverse complements. I think the idea of the question is that, one, error correction is at least in theory feasible for genome assembly. But secondly, even for this simple problem, there's quite a lot to do because you have to handle reverse complements and so forth. So this won't necessarily be a very straightforward question. What we're looking for is, given a collection of sequencing reads, represented as a list of strings, we want a bunch of tuples. So we're going to have a list of tuples. And what are each of those tuples? We have the incorrect read and the corrected read in the data set. So in order to write this function, well, the first thing that we're going to do is let's simplify things a bit by, instead of having to go through and figure out where the errors are straight away, this is sort of, when somebody comes to the problem, if you haven't solved a lot of problems before, you might be worried to think, I need to find the errors. And really the first thing that you need to find is, why don't we just go through the data set and identify the counts of everything. And so let's first let's identify the number of occurrences of all our reads with reverse complements. And I'm going to pass all that work to a subroutine. So I'm going to have a subroutine that's going to give me a dictionary, and it's going to be called count with reverse complement. And it's going to take reads. Okay. And we can see that subroutine here. So it's going to return a dictionary. Mapping strings to integers. And this is our reads to integers. And this is number of occurrences. Once we've got that, we're going to say, all right, which of those are erroneous? Now we can say, so I've assumed like the ideal version of what I would really like, which is each one of our reads is going to correspond to an integer telling me how many times that I see it. Let's go ahead and write that subroutine. I think that's a good subroutine to write. So we're going to take a list of DNA reads. We're going to return this dictionary mapping each read to its, we say, combined multiplicity, meaning the number of times it appears plus the number of times its reverse complement appears. I will make a raw dictionary first, and that raw dictionary is going to be a dictionary mapping strings to integers, and it's going to be empty, and I'm not even going to handle reverse complements. So the first thing we're going to do is, let's first just identify the number of occurrences of each string. And to do that, I've got my raw dictionary that I'm going to range over the reads, and I'm going to set raw var is equal to raw.get. And what I'll say is, I want to access what its current value is. If I've not seen it thus far, though, I want to set it equal to 0. Regardless of whether I've seen it before or not, I want to add 1 to its current value. So raw.get of r0 is going to say, is r in the dictionary as a key? If it's not, create a value of 0 associated with it. If it is, grab its value, so it might be 17, and in either of those cases, we're going to add 1 to it. So this is a common way of writing this idea. I'm ranging through a collection of things, and I'm counting up the number of appearances that I see. It's a very standard practice to just set the key equal to what its current value is plus 1, and if it doesn't exist, set it equal to 0, which is where this .get comes from. Okay. Once I've done that, we're going to set all the counts, including the reverse complements, using a separate dictionary. Okay. So to do that, I'm going to say, let's have a counts dictionary. It's going to be a dictionary mapping strings to integers. It's going to start off as empty. That's the thing that I eventually want to return. And in the meantime, well, what I want to do is to say range over the reads and identify the reverse complements. So for R in reads, I'll call RC the current reverse complement of R. Fortunately, that's a function we've already written. So that's imported from revc.py. That's one of our first questions on the platform, and now we get to use it here. Once I've reverse complemented the current read, I'm going to say, well, look at counts of R. And counts of R needs to get set. So as the sum of its raw of R and raw of RC. So I've got the read, I've got its reverse complement. The number of times I've seen R or its reverse complement is equal to, we're going to say raw.get of R 0, because really we would love to say raw of R plus raw of RC. But if I take this sum and they don't exist, right, like, well, we know that raw of R is going to exist, but it might not be the case that raw of RC exists, because its reverse complement might not be in this raw dictionary, because we saw an error there. So what I'll do is I'll make both of those a raw.get. So it's R0 plus raw.get of RC and 0. And that's it. So the counts dictionary that I really want is now set. And that's nice, because once I have that dictionary, now I can go back to the function that I want to write. I understand what it's going to say, well, which is which of these reads in the data set are erroneous, look at counts, and find the elements that don't have a value that's bigger than or equal to 2. So I'll range through the counts, and then I'll say, okay, first off, I'm going to form, we're going to range over the reads. And I'm going to say, is it the case that counts of R for the current read R is bigger than or equal to 2? And if it is, well, it's correct. And otherwise, it's incorrect. And I'm going to do something as simple as saying, well, I'm going to maintain 2 sets. I'm going to have a correct set, and I'm going to add R to it in this particular case. And Python's angry at me because I haven't declared correct. And in this particular case, it's incorrect, so I'm going to have an erroneous list, and I'm going to append R to it. I think the idea here is we're not going to double add anything to the correct set. So I'll say correct is going to be a set of strings, and it's going to start off as an empty set. Erroneous is going to be a list of strings, and it's going to be empty to begin with. I think that's a minor point, that this is a set and this is a list. Once I've divided them out, that's super helpful, because now I know what the correct ones are and what the erroneous ones are. And once I know that, now I can just make a list of the corrected reads, plus their matching partners, or I should say their mismatching partners. And so I'll say corrections is a list that we want, and the list that I want is this list of tuples. So it's a tuple of strings containing two strings, so it's a tuple of string, string. It starts off as an empty list. And then what I will do is range through it and say, let's go through the erroneous list and find partners. So I will say for, let's call it bad string. This is the one that we know is either it has an error or its partner has an error. We'll range through it, and then we say, well, let's find a matching partner. So I'm going to say find matching partner equals, I'm going to have a subroutine to do that called find match. And I'm looking for this the matching partner of the bad string in the erroneous list of strings that I made. So this is the collection of strings that we know are not correct, or either, I should say, maybe they're correct, but their partner is incorrect. Either way, they're going to fall into the same bucket. So I'm looking through that collection of strings to find the matching partner of bad string. And once I've found it, that's good. So once I find a match, append it to the corrected set. Should say list. And so we're going to say corrections dot append the bad string and its matching partner. And at the end of this whole process, I'm going to return corrections. Now we're making a strong assumption here. I guess technically what we might do is to say if, as a result of this subroutine, if it's not the case that it's none, meaning, I actually did find something, only then will I append the two things to each other. So I'll say, I'll append this tuple. But the data set that we're going to have has the guarantee that we're always going to be able to find the matching partner associated with it. Then it is just a matter of saying, let's write the subroutine to find the match. So we need a find match subroutine that takes this bad string and it takes the correct set of strings and find something in this set that differs from the bad string by exactly one nucleotide. And that could include reverse complements as well. Remember, differing by one nucleotide means that I'm going to need to import the Hamming distance from HAMM.py, another introductory problem that we solved long ago. And so that's going to be useful to us here. And so when I write find match, what I want to do is to say, well, for every good string in this correct set, if it's the case that I access the Hamming distance between the bad string and the good string, let me call this bad string, just to be completely clear. And if that's exactly equal to 1, we don't want it to be less than or equal to 1. That would be dangerous because then we might be finding a match. So if it's exactly equal to 1, which is what the particular problem is asking for, then I want to return good string. Okay, so this is the string that I want to return, which is my matching partner. If I ever get down here, this is where I'm going to return none. So this is our default. We didn't find anything. The other thing I want to keep in mind, though, is, we need to check reverse complements. So I've got my good string, and let's take the reverse complement of it. So I get to call reverse complement again and access good string. And now I say, is it the case that the Hamming distance of RC good string with good string. I guess bad string. And I can do that in either order. I think this makes a lot more sense if I switch this with this, just so it's consistent with the previous call to Hamming distance. Okay. And if that's the case, so when we look at our set and we access the reverse complement 2, if the Hamming distance here is equal to 1, then I want to return the reverse complement of the good string as the partner. And that should do it. So we have our sample data set. We've written all of our functions here. We've got our read set from Rosalind. The order may vary on what we actually find, but something like this, we should see three examples. And so we can run this, make sure we're saved, and then we're running Python 3 corr.py, and nothing happened. That's not good. Seems like nothing printed. It's like maybe I didn't find anything. Okay, that's bad. That's not good at all. So we can see that we've got some kind of bug here. Great. Yeah, length of corrections is 0. That's not good. Well, well, well. I definitely found that issue. Okay, that's not going to fix it. Let's take a look. Take a look just before we dig into the details here. So count with reverse complements of reads. So far, so good. I think these are good. That's good. Incorrect. Erroneous. Yep. Thing looks okay here. I'm in the wrong language here. Print counts. See what happens. Okay, so this looks pretty good. So just to take a look at our dictionary, TC occurs twice, this occurs once, this occurs twice, once, twice, 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 once. So each of these is the reverse complement of someone. And with reverse complements, they should occur twice. So we have 1, 2, 3, 4, 5. To say this is a little worrisome. Because there's only 3. Right, in this particular case, TTGAT, that is one away from TTCAT. And this occurs once. TTGAT occurs once. And its reverse common would be ATCAA, which also occurs here. Yeah, so this is a very precise data set. This does appear good though. Okay. So I'm thinking it's not in that subroutine. The other thing we might try is, let's see, when we call find match, let's see, I don't need this. Let's see what happens down here. Double check this. So 2, 3, 4, 5, 6, 7, 8, 9. We had 9 input strings, I believe. Yep. Okay, so the raw dictionary looks good. Okay, so that seems fine. Print correct. I think that's what we found before, is the things that contain 2. Yeah. So everything there looks good. And erroneous is those 3 strings, which is what we found before. So far, so good. Everything there looks good. So we're narrowing this down pretty good. If anybody in the chat wants to exude some brilliance, this is going to be a very annoying little thing. So looking for all the bad strings in the erroneous set. If we find a match between the bad string and I think this is our problem. So when we find a match, we're not looking against the erroneous set, we're looking against the correct set. So that should do it. That should do it. List dot append takes exactly 1 argument. What would... Yeah. So we never actually made it here. Okay, I gave it 2. It's that old tuple issue with Python, which is that I'm appending a tuple. So, yep, I think we're good. Oh, here we go. All right. So Joel let me flounder here for like 7 minutes, and then type yes, line 43. I hope you didn't know that all along. Anyway, we still might have a wrong problem, but let's see what happens. Let's comment this out. Or uncomment it out, I should say. And then we're going to get a data set. Okay. Let's look at our output. All right, we got it. All right. A little real-time debugging with no test data sets. That's always fun. Joel, you saw it at the same time as me? Yeah, sure you did. Sure you did. I believe you. Okay. So So we got 3 more problems to go today. And I'm going to give you a hint. They're not going to take as much time probably as the 2 that we've already done. But we're going to move on. A couple tree problems. Okay. Our next one is TREE. Get it ready for us. All right. TREE is completing a tree. And I'm going to show you my favorite 2 evolutionary trees. I think they're the 2 most important evolutionary trees of all time. First one is from the mid-18th century, Darwin's Notebook, the first known evolutionary tree ever to be drawn. And the second one on the right, I remember seeing it for the first time. It's from 2016. And this is a simplification of that tree, because it's not showing all of the individual tiny little clades labeled, so it's like a high-level representation of it. But it corresponds to sequencing the same gene in a huge number of different organisms, feeding this into a big algorithm, and seeing what comes out. Why I like it is Darwin's thinking about evolutionary trees as the giraffes over here and the zebras over here. What this tree on the right shows from Hug et al. Is, well, the tree really is a huge collection of bacteria, some archaea, and then we humans within this tiny green sliver of the eukaryotes. So I always tell my students, this is really a bacterial planet that we happen to inhabit. From the perspective of genetic diversity, the bacteria overwhelm us by a large margin. This is a biological notion of what an evolutionary tree is. But if we're going to work with algorithms, we need a quantitative, mathematical, computational notion of what a tree is. And if we ask the mathematicians, they'll give us the best, cleanest, simplest explanation for what a tree is. They say a tree is a network or a graph, 2 terms for the same concept, that is connected. Connected meaning it's possible to have a path of edges between any pair of nodes. So pick any 2 nodes in the tree. It's possible to reach one node from the other, but it also has no cycles, meaning it's not possible to have a collection of edges that form a path or a walk through the tree that returns to the node that you started with. So you can see each of these 3 graphs that I've shown here satisfies both of those conditions. The connected condition, meaning you can reach every node from every other node, is important biologically because it means every species that's represented in this tree, that are shown as the darker nodes on the leaves on the outsides of the tree, come from some most recent common ancestor. And the acyclic condition is important because it means that there's not like horizontal gene transfer going on that's exchanging genetic information from one part of the tree to the other. And that's a strong assumption. There are certain types of trees, especially with bacteria, where there is horizontal gene transfer. If we're working with eukaryotes, though, the evolutionary tree is a great construct. As I mentioned, there's kind of 2 types of nodes that are colored differently in this figure. So you have the darker colored nodes correspond to leaves. They have degree equal to 1. What's the degree? It's the number of edges that are connected to it. So you'll see every single one of these darker nodes, and each of these trees is at the ends of the tree, and we can clarify and quantify that by saying they only have one edge that they're connected to. All of the other internal nodes correspond to ancestral species, where these darker nodes are like present-day taxa. The lighter nodes are ancestral species that are not necessarily in our data set, but that represent places where there was evolutionary branching. The idea is this might look more like an evolutionary tree to you than this, but from a mathematical perspective, they're both trees. But why? Because they both satisfy the definition of being connected and acyclic. The problem that we're going to have is, if we look at Rosalind, our issue here is with this data set, we're going to say, well, our tree is missing a few edges. It's not quite a tree yet. And it's not a tree. We actually can draw this out because it's not going to be connected. So our first input in the data set is going to be the number of nodes. Everything else is going to be the adjacency list, and we've worked with adjacency lists previously, but it's a collection of node 1 connected to node 2, node 2 connected to node 8. So these are representations just with pairs of integers that tell us what the edges in our network should be. So what we want to output is, what's the minimum number of edges I need to add to this network that will allow me to have a tree? And there's two ways of solving this question. So if I can actually take, and I'll show you, our sample data set is, let me just copy it over. It's this. Okay. So that's our sample data set. We know that there's 10 nodes. And I can go through and start to infer, what is it that the edges look like? And I can say, this tree is going to say, well, I've got 1 connected to 2. That's good. I also have 2 connected to 8. And 2 is not connected to anything else. And 8 is not connected to anything else. So that's one what we would call component of this network. We have 4 is connected to 10. And we have 10 is also connected to 6. So we see 2 nodes, 2 edges there. And then we have 5 is connected to 9 and 7 is connected to 9. And then that seems like we're finished, but we're actually missing 3 entirely. It doesn't have any partners. So then we have 3 all by its lonesome self. And I don't know why I did this way. If 9 is connected to 7, we can just do it like this. So here are our components. So we have 4 components. What are components? They're subnetworks that happen to be connected. And I could join these connected components into a connected network that would not be cyclic if I connect any node from here into any node from here, and any node from here into any node from here, and then just connect 3 into wherever you want. So as you can see, you have a lot of choices for what these could be, but the minimum number of edges that turn this is called a forest, into a single tree is 3. Why? Because there's 4 components, and you pick 3 edges to connect those components. So that's all well and good. That's why we get an answer of 3 for this particular question. If you only have 2, well, every edge is going to join 2 components together, so you're not going to have enough edges to join the 4 components into a single one. And this gives us there's kind of a natural way of solving this question. We could first form all of the components of the network. So we could first figure out that there are 4 connected pieces in this forest. And then we could say, well, if there's 4 connected components, I need 3 edges in order to connect them. All right, so you would just take the number of components and you would subtract 1. That means we would need to write a depth first search algorithm that'll start at a given node and try and explore and say, connect everything that we can in that component. And then when you run out, get rid of those nodes, look at the next node, depth first search on it. And the problem reduces to finding connected components in a network. So this network is kind of separated out into these separate pieces. How is it that we would actually figure out what those pieces are? But the smarter way is to use a little bit of math. And the little bit of math is based on the idea that although these trees come in all different shapes and sizes, they all share something. What they share is there's a relationship between their nodes and their edges. So in all of these networks, all these trees, if I count up the number of nodes in them and I count up the number of edges always happens to be equal to the number of nodes minus 1. And there's a theorem behind the scenes. So there's a beautiful invariant mathematically that tells us if I have a tree, meaning a connected acyclic network, it has n nodes, it always has to have n minus 1 edges. There are two conflicting forces here at work. You have the connectedness means you have to have some amount of edges in the network. So that boosts the number of edges. And then you have the condition that this network has to be acyclic. It can't have cycles in it. Cycles would mean there's lots of edges. So connectedness means there's enough edges. Acyclicness means there's not a lot of edges. And when those two competing forces balance, for whatever amazing reason, we can prove definitively that if this is a tree, connected acyclic network, it always has this invariant property that the number of edges is equal to 1 less than the number of nodes, which is pretty cool. It's beyond our work here to show that proof. It would take us about 30 minutes, an hour or something to go through it. It's really nice, and you can have a concrete explanation for why this is the case. In our case, well, we can use this very much to our advantage because if we look at our data set, we know that there's 10 nodes in our tree. How many edges are there? 1, 2, 3, 4, 5, 6. How many edges would there need to be in order for there to be a tree? There need to be 9. So in this case, we have 10 nodes. We have 6 edges. And the number of additional edges to form a tree is 10 minus 6 minus 1. Why? Because, well, number of edges in a tree is 10 minus 1 is equal to 9, and so it's just 9 minus 6, which is equal to 3. And in general, we're going to have n nodes, we're going to have k edges. The number of edges in the tree is n minus 1, and the number of additional edges is going to need to be, well, n minus 1 minus k. And so our function, which is going to take the number of nodes, and it's going to take this list of edges, and thank goodness I'm giving you all the parsing code, so we don't actually have to parse out the data set, right? But it's just a list of tuples of pairs of integers. At the end of the day, all I have to do is say, what's my idea? Figure out how many nodes there are, figure out how many edges there are, and then just return this formula. So the number of nodes that I have is number of nodes. The number of edges that I have in the desired network is that minus 1. And we take the difference between that and the number of edges that I have, which is the length of edges. So at the end of the day, all I have to do is return that thing. And it's a one-line function. And we can run our code by saying Python 3 tree.py. We get an answer of 3. And I'll uncomment out my parsing and get a data set. Call it data set. And run this thing again. And then we have a big data set here, right? It has 883 nodes in it. Here are all the edges here. But at the end of the day, when we count this up, we take 883 minus 1, subtract the number of edges, and for this particular data set, well, the answer is 88. Could even just type that in. And fortunately, the maths. So that question really, that was me being as tricky as I could be. That's just me being mean and saying, if you know this fact, well, then it's going to be very helpful. It's going to be very helpful for solving the problem that you want, because this is an easy question in disguise as a hard question. We could go through and do a bunch of algorithms here, or we could just apply a little bit of math and know that there's 3 missing edges and be able to figure out, well, I could even actually know that regardless of how these edges are connected, I know that there's 4 connected components. So that's pretty good. Let's see if that idea is an isolated occurrence, or let's see if I've been tricky in other ways. So we're going to look at INOD as our next one. And I'm just going to get us ready to do it because I'm going to move straight into it. All right, INOD is our next one. All right. Okay. Yep. So we're ready to solve these next two. All right. INOD is counting phylogenetic ancestors. It's a problem that builds on what we've seen previously with trees. Remember, what do we mean by a tree? A tree is a connected network that also doesn't have cycles, or is acyclic, as we say. We also saw that trees can have beautiful properties to them. So any tree that has n nodes has n-1 edges. It turns out that's a hint to this question. So to say, you may have not known that fact. You might have figured it out as a result of solving the tree problem. And it turns out that this is going to be useful in this particular question. So we're going to work with what are called unrooted binary trees. So we have this theorem that every tree with n nodes has exactly n-1 edges. Some trees are rooted. So here's a tree of a collection of related organisms where we designate one of our nodes as the root, and then time flows downward from the root to the leaves. We're going to work with unrooted trees in this particular question. So they're trees that have a special property. They don't have a root in them, but they're called unrooted binary trees. And what this means is we have two types of nodes. We know that trees have two types of nodes. We have the present day species are assigned to what are called leaves. They have degree equal to 1, which means that each one of these is at the end of the tree and has only one edge that connects it into the rest of the network. We've also got internal nodes, and in an arbitrary tree, those could have 15 edges connecting them, but in a binary tree, they only have 3. So this means that regardless of which direction I travel in the tree, every time I hit an internal node, I enter it with one edge, and then I've got 2 leaving it. So I've got 2 leaving it that way, and I've got 2 leaving it that way. If I go in this direction, I have 1 edge in, 2 out, 1 edge in, 2 out. So that's a strong property, but a lot of evolutionary tree algorithms will produce a binary tree because it's based off of this assumption that, well, regardless of which direction evolution is traveling, every time I encounter a new internal node, there's a speciation event that divides it into 2 other species. And sometimes that divides it into 2 present-day species. Sometimes it divides into a present-day species and another ancestor, or into 2 ancestors and so forth. So we're working with this special type of tree structure. And the idea here is, well, we're going to be given a positive integer n. And in keeping with this idea that there's an invariant for trees in general, that if you have n nodes, you always have n-1 edges. What this problem is suggesting is we're not going to trick you. We're going to give you this strong hint that there's another invariant here, which is that if I have a positive integer n, there is an invariant that says regardless of how I connect all the leaves together into an unrooted binary tree, we know that the number of internal nodes is going to be somehow a function of n. So for any unrooted binary tree with 4 leaves, the number of internal nodes, ancestral species, if we have a tree like this, is always going to be equal to 2. So it's making that suggestion that there's an invariant. It doesn't have to be true, right? One of the things about math is that sometimes you might think that there's a rule underlying something, and it doesn't turn out to be the case that there is a rule. But I'll give you a hint that we wouldn't have written the problem in this way if there weren't an invariant in this particular case. So just as it didn't have to be the case that any tree with n nodes has to have n minus 1 edges, there's going to be another invariant here that says if you're an unrooted binary tree and you have n leaves, you're going to have some function of n that's going to say if I have n leaves, how many internal nodes do I get? All right, so the point of the problem is to return that number of internal nodes as a function of n. The idea of why there's an invariant is that, well, demanding that all of these internal nodes have degree equal to 3 is a pretty strong assumption, right? So, you could imagine a different tree where there's a node here in between these two and it has degree equal to 2. That's not an unrooted binary tree. Or you could have, a different structure where one internal node branches out into 4, and that's also not an unrooted binary tree. So it's a strong assumption. The hint says that n nodes means n minus 1 edges in any tree is important. We probably should use that. And what's going to tie these together is something that's called the handshake lemma. So the handshake lemma says, if we consider a party where n people shake hands, and we say, well, they may pair off in a lot of different ways, but we count two different things. Count one is ask everybody how many hands they shook and add them. Okay, so we go to every single person, we ask them, how many times did you shake somebody's hand? And somebody says 0, somebody says 12, somebody says 2, and so on. And add up all of those numbers. So that's the sum of all of the times each person shook a hand. And count 2 is the total number of handshakes. So we have a camera that records the footage and says, all right, well, I was able to count how many times hands were shaken. If everyone is honest, then I don't think it's tremendously hard to see that there's a relationship between these two things. Every time somebody shook somebody else's hand, that corresponded to a handshake, except there's two partners as a part of this. So every time somebody added their sum of handshakes into the pot, there were two people that contributed to that, each person on each side of the handshake. As a result, well, we get that count 1 is equal to 2 times, not 3 times, 2 times count 1. And that's an observation about a party, right? But at the end of the day, it's an observation about networks as well. So if we go back and we look at a network like this, each edge in our network can be thought of, and this is for any network, can be thought of as like a handshake. So the number of edges is our count 2. That's the number of handshakes at our party. Well, what's count 1? Count 1 would be to go to each node and say, how many hands did you shake? So to say, what is your degree? So look in all of the different directions that you can see. So in this particular case, well, everybody is going to shake 1 hand or 3 hands, depending on if you're like our orangutan buddy here who's on a leaf of a tree, or if you're at an internal node, you're going to shake 3 hands. So if we take that idea, what it means is, in graph theory, the handshake lemma says count 1 is the sum of the degrees of all nodes, and count 2 is the sum of all edges, or the total number of edges. And by similar logic, count 1 is equal to 2 times count 2. So far, so good. So this is why it winds up being very useful. Okay, so this is our handshake lemma that says the total number of edges is equal to or 2 times the total number of edges, is equal to the sum of the degrees of all the nodes. Remember, degree equals number of edges incident. So how does that apply to this particular context? Well, let's think about, in the case of trees, we know that if there are n nodes, there are n minus 1 edges. Right? That's a fact that we have from the previous problem that we solved. But we have something that's even stronger than this because, well, we have an unrooted binary tree. All nodes have degree 1 or 3. So that's the assumption of what we're working with. So what we're going to say is assume that we have n leaves in an unrooted binary tree. Okay? And say that there are k internal nodes. So I don't know how many there are. We want to find a relationship between k and n. It might not exist, or it might. But what we know is the total number of nodes is k plus n. So the total number of edges is k plus n minus 1. Why? Because it's a tree. So k plus n minus 1 is the total number of edges. But then, well, what about our handshake lemma? The handshake lemma says to sum the degrees of the tree, of all the nodes. Okay? And, well, all the nodes fall into 2 categories. So each node could be an internal node. Remember, what's our definition of an unrooted binary tree? You could be an internal node and have degree 3. You have to have degree 3. Or you can be a leaf and have degree 1. So it's not actually hard to get a formula there by saying, well, the sum of the degrees is equal to the number of leaves, which is n, and each one of those has degree 1, plus k, which is the number of internal nodes, times 3. So it's really just, and let's write that in a little bit nicer of a way, n plus 3k. And the handshake lemma also tells us this sum is equal to 2 times the number of edges. So anytime things are equal in math, we probably are going to have to do a little bit of algebra and write an equation, and we're going to say n plus 3k is equal to 2 times the number of edges that we have. We already have a formula for the number of edges. We reduced it to k plus n minus 1. So we're going to say 2 times k plus n minus 1. And once we have that, we can expand the right side. So we can now say n plus 3k is equal to 2 times k plus 2 times n minus 2. Just a little bit of distributing that 2 across the parentheses. And once we have that, we do have to do a little bit of dreaded algebra, but it turns out to be pretty easy because this n subtracts out and goes over here. We get 2k going over here. So at the end of the day, we get k on the left, and we get an n minus 2 on the right. And lo and behold, here's our relationship. What was k? That's the number of internal nodes in our unrooted binary tree. What was n? It's the number of leaves. So here's our formula. And at the end of the day, if you give me the number of leaves in an unrooted binary tree, and you want to know the number of internal nodes, all I have to do then is just return num leaves minus 2. Could it be so easy? Certainly passing our test. Comment the parsing part of the code. And let's put our data set here. Call it data set. Run her again. We get 7138. I think I can remember that. I'm guessing that the data set was 7140. And we get the problem right. So the maths once again. Now, 3,400 people solved that. Did 3,400 people do this and understand this? Like, did we have 3,400 handshake lemma stands? I doubt it. I would love to believe that. I really would. I also think there's a possibility that by the 20th time you tried the problem and you saw that the output was equal to two less than the input, maybe you came up with a conjecture and just got it right. But either way. We're going to have one more for today. It's funny. There's going to be more tricks. There's going to be more tricky stuff. I just didn't really have the heart to do another tricky one. I feel like it might make sense to have that kind of maybe appear in a later time. But we're going to look at PDST, which is just going to set up some of our thinking for preparing us for more work in what we actually want to do with evolutionary trees. All right. PDST is creating a distance matrix. We started working with evolutionary trees, and we started seeing some properties of evolutionary trees and the fact that sometimes they're going to be properties that always have to be the case around these trees. I love the math side of things, and I could do the tree math all day long. But ultimately, there is a point of using a tree, which is as a biological model. And what we want to move towards is constructing an evolutionary tree from biological data. So what we really want is to have some way of representing connections between species or taxa, and then form a tree from that. So regardless of how we form this data set, a classic way of producing an evolutionary tree is to have what we call a distance matrix. And we could talk about how this distance matrix gets formed in a lot of different ways. You could check out bioinformatics algorithms where we talk about multiple sequence alignment and so forth. But in whatever manner that we obtain the distance matrix, we have a distance matrix representing pairwise distances between our collection of organisms. And then what we want is a tree that explains that. What does it mean for this tree to explain it? Well, the present day species need to be on leaves of the tree. That makes sense. And perhaps we weight edges of the tree that we form in such a way that when you sum the contributions of all those edge weights, it matches what happens in the tree. So what I really want is a tree that fits the distance matrix. The distance matrix has its collection of values that are pairwise, comparing human to seal and whale to chimp and so on. And you'll notice that it's symmetric across the main diagonal because, well, the distance from any organism to itself is 0. And the distance from human to chimp should be the same as chimp to human. But the idea is when we take the sum of the edge weights in the tree, hopefully that would match what exists in what we may call capital D, which is our distance matrix. So, for example, the distance from chimp to human in the tree is 3, 1 plus 2, and it's 3 in the matrix. It's 1 plus 3 plus 2 in the tree, which is 6, and it's 6 in the matrix, and so forth. So, for any pair of organisms, the distance between them in the matrix is equal to the distance between them in the tree. You're given the matrix, and then you want to form the tree. That's our central question that we want to look at, which is, I've got a matrix, how do I form the tree from it? That is the subject of another day. That's a huge problem. We could have textbooks on this, and it's the subject of work for decades. You can, again, check out bioinformatics algorithms if you want an answer for now. But what we're going to do is, well, let's start thinking about how you might form the distance matrix. So this question is just going to be a nice little warm-up that's going to say, we're going to have a collection of strings as input. They're all going to have equal length because we're nice. And then we're going to form a simple distance metric that's going to allow us to compare each pair of strings so we get comfortable with representing distance matrices. One simple thing that we could do is to just take the Hamming distance between pairs of strings. We're going to do one baby step beyond that, which is to take the Hamming distance and divide it by the length of the strings so that we're at least normalizing by the length of the string so that we can make a comparison that doesn't matter how long the strings are. But that if you compare this string against this string, they have some similarities, but they're going to differ in 4 positions. So the first 2 positions, they're going to differ. And then this position, they differ. And this position, they differ. So they have length 10, and they differ in 4 positions. So that's how we get this value and this value equal to 0.4. We're just dividing the Hamming distance by the length. So this against this is very close to each other. These 2 strings are similar. They differ only in the first position. And so this element of the distance matrix should be 0.1, and this element should be 0.1 because they differ in 10% of their positions. So we define this make-believe distance called P distance, which is just taking the Hamming distance and dividing it by the length. We could come up with, a way of forming the distance matrix in a much fancier approach. We've already seen something like edit distance that we could use to compare pairs of strings. So we could already do something that's fancier than this, but the purpose of the problem is just to get us comfortable with seeing this concept of a distance matrix. So we're going to have a P distance that I'm just going to go ahead and write because we've written Hamming distance in a previous problem. So we know how to count the number of mismatches between 2 strings. We've got that imported. And if we've got that imported, I'm just going to return the Hamming distance divided by the length of S. Now, or the length of T, which means I probably should have a check that says, look, if they're not equal, let's be precise. I know we're in Rosalind problems and they're always with very specific data formats, but I should raise a value error and say, the strings have differing lengths. But if I make it here, I know they have the same length and we're good to go. What I really want to do is make this distance matrix. And I'm going to say, well, first off, I've got a collection of sequences. And then I want to make a distance matrix, which is a 2-dimensional list of floats. And regardless of whether I'm using P distance or edit distance or something else, it doesn't really matter. I'm going to say that the number of sequences is equal to the length of the sequences. So that's important. So now we make an n by n matrix. And we should set the main diagonal values to 0. And so to do that, I'm just going to say with everybody equal to 0 at the start. So I'll say 4 underscore n range over the number of sequences. Before I even do that, let's say the matrix is a list of floats. And it's empty to start off with. But then I'm going to range over all of the sequences. And I'm going to make a row of length n. And so row is a list of floats. And it's just going to be 0.0 times n. And then I'm going to append it to the matrix. Okay. So once I've done that, at the very end of this, I'm going to return the matrix. And in the meantime, well, we're going to range over all the values that we want to set and call the subroutine. So I know how to range over a 2-dimensional list. I can say for I in range over num sequences. And for j in range num sequences. And as I do that, well, I want to say, what's the current distance? I'm going to call that d. It's the P distance between the current strings that I'm working with, which is sequences of I and sequences of j. And once I've determined that distance, great. I just want to set it equal to matrix of ij. So there we go. And this will work. I can run this. I think we've got a test. Okay. It needs to make sure there's one important thing, which is that when I run Python 3 PDST.py, name n is not defined. That's not good. Where did I? At times num sequences. Okay, let's try that again. Unsupported operand types. I'm really leaking oil here. All right. I got so ahead of myself that I forgot to say it's the Hamming distance of something. All right, let's try it one more time. All right, and now we get what we want. And it appears to be correct. We can punch this into Rosalind for a larger data set. But this function could be a little better. And the function could be a little better because, well, you think one thing that we're doing, we're computing the p distance between I and itself. So if I is 3, for example, well, j is going to range between 0 and the number of sequences. So j is eventually going to be equal to I. And, one of the things that we could do is say that's kind of pointless. So if j is not equal to I, then we would do this. Okay. So we don't need to compute it in that particular case. That's one way of getting around it. That's perfectly reasonable. The other thing that we should probably notice, though, is that this value is equal to this value. Of course it is. This value is equal to this value. Because if you change the order of the strings that are given into the p distance, it's just counting mismatches and dividing by the length. So we should have a symmetric distance matrix regardless of how that distance is formed. And so it seems weird that, well, we not only don't need to set those values on the main diagonal, we also only need to compute like this value once, and then that value could fall out from that. And so I don't need to do it this way. What I need to do is to acknowledge that my ranging is just doing too much work. These values have already been set to be equal to 0. Why? That happened here. Okay. And so really I only need to look at these values and these values and these values. So what I can do is to say, I'm going to range up to the number of sequences, but I'm going to start at I plus 1. Why? Because, well, when I is 0, I want to start j at 1. When I is 1, I want to start j at 2. When I is 2, I want to start j at 3, and so on. And so that will allow me to access this upper right part of the matrix. Now, if I run this, I want to be very careful, because what will happen is I've set the upper right part of the matrix for my test, but I didn't get the bottom left part of the matrix. But here's where the symmetry is helpful, because if I know this value, I get this value for free. If I know ij, I get, well, ji for free. So I'm going to set matrix of ji equal to d. That makes my algorithm about twice as fast because I only compute d once. And now I get my correct answer, which means I should be ready to uncomment out my parsing code, get a data set from the platform, put it into the file explorer and name it dataset.txt. And run it again. And now we get a longer answer, but it goes to output. And I can just copy that over. And we get a correct solution. Plus, we get our alignment badge. Okay. We're in a pretty selective club of fewer than 1,000 people globally. And when we look at our list, see we've solved a lot of problems now, and we're getting into the rarefied air. So we have one. We need to come back and do KMP. This problem has 3,000 solutions, but now we're ready to, start solving questions that only have a few hundred answers. And now I think after we do a few more, we're going to start having problems that have under 1,000 pretty exclusively. So we're getting there, and we're not that far away from a collection of problems that only a couple hundred people have solved. So we've done a lot of work. Now that we have all this foundation, it's going to help us continue to take one step at a time and get towards more advanced questions. With respect to alignment, we're going to work a lot more with these distance matrices later, and evolutionary tree construction a lot more later. Regardless of how we make the trees, or how we make the matrices, I should say, the question is, how do we best form a tree from them? And that's a complicated question with quite a lot to it. As such, it's the subject for another time. I can't wait to see you then. I hope you'll join me then. And until then, all the best.